Thank you, Sylvia, for this wonderful presentation, and congratulations for your awards. Hi, everyone. My name is Romina Schiaffino from CONICET, Argentina. It's a great pleasure to introduce Hilary Close, Associate Professor in the Department of Ocean Sciences at the University of Miami Rosenstiel School of Marine, Atmospheric, and Earth Science. She specializes in marine organic geochemistry and the ocean carbon cycle. Her research focuses on how microbial processes and particle dynamics regulate the production, transformation, and export of organic matter to the deep ocean. She's an expert in the development and use of compound-specific isotope analysis. Her work has advanced our understanding of the biological carbon pump and its role in long-term carbon storage and climate regulation. The plenary of her presentation is Hidden Beneath the Surface, Unveiling the Distinct Biogeochemistry of the Lower Eutrophic Zone. Please join me in welcoming Professor Hilary Close.
Okay, thank you so much for this opportunity to the organizers for giving me the opportunity to fill in for my colleague Max Saito. This will be a different topic. And I am switching to the ocean for most of this talk, but I do want to draw connections to lake systems that I think exist with some of what we're looking at. And I will actually start by just mentioning my pathway through science actually started here in Northern Ohio. I'm going to mention this for two reasons. One, it has a maritime border with Canada, which I never even thought about until I was getting a security clearance for something. And number two, I grew up on a family farm, and our drainage drains ultimately into Lake Erie. So I am really happy to talk with anybody working on lake systems and eutrophication. I'm still closely tied to our family farm there. And then, so those are the places that I have trained, and then these are the places that I have been able to conduct oceanographic research. And so I'm really accumulating the global ocean a little by little over time. And this is also a way of introducing one of the main ways that we can take a snapshot of the global ocean is by looking at satellite chlorophyll imagery. So I'm going to start there to introduce our main environments for this talk, which are the oligotrophic gyres. And I'm going to specifically be talking about the time periods of the year in which those gyres are thermally stratified. And the euphotic zone develops quite deep. So oftentimes we see that 0.1% surface irradiance reaching down to about 150 meters. And just in case anybody isn't thinking about this frequently, we do think of the euphotic zone as that part of the water column where photosynthesis is occurring at measurable rates. And so I won't be going beneath the euphotic zone very much today, but I'm happy to talk about that later. And so, of course, our ocean gyres, our subtropical gyres, are the main oligotrophic environments throughout the global ocean. And I have been making measurements in 3 of those gyres, one of them very recently, but I'm going to focus most of what I talk about today in the North Atlantic subtropical gyre off of Bermuda. And, but we are expanding these measurements, and we're starting to see a lot of coherence across these. Okay, and I'll try to mention as I go along why thinking about these environments and the patterns that I'm talking about are important. And one of the big things is that these oligotrophic environments are expanding as sea surface temperatures rise because of the increasing thermal stratification. So like I said, one very useful way to look at these environments is by satellites. We're looking here at annual average chlorophyll. And I want to start with the question is, what is it that satellites are actually seeing? And so we can look at a bit of a schematic of biogeochemical cycling in the ocean. And this is really just to point out that satellites see the surface. And so satellites now have data retrievals and various algorithms that have been developed that allow us to make a lot of estimates about the types of phytoplankton communities that are active and also some of the food web cycling occurring. Nonetheless, the observations are only in the upper few meters. And as I just said, in a lot of these environments, the euphotic zone can extend down to 150 meters. So clearly, this method is not going to directly capture all of the relevant primary production happening in the upper ocean. And so this was really the focus of the exports program, which was a program funded by NASA, and I was involved in it through partnership with the NSF. And this is one of the big diagrams from the exports program showing the complexity of both production and of recycling processes in the upper ocean, and then how that translates into what gets exported into the deep ocean and stored in the deep ocean or consumed, or potentially getting all the way to the sediments. So the idea behind exports was, in fact, to be able to better essentially ground truth what we're trying to predict from satellites by making very intensive in situ observations. Nonetheless, we are at a point that we still need to make in situ observations to really capture processes below that upper layer. So that's what I'm going to be focusing on today, is sort of the complexity that we can capture through those in situ observations. Okay, so in case you have not worked in oligotrophic systems like this, like I said, the euphotic zone tends to extend to around 150 meters, sometimes deeper. This is some data from the Bermuda Atlantic Time Series study site, and this is just showing some very basic properties of these types of water columns. Number one, our photosynthetically available radiation steeply drops off as the light is attenuated through the water column. And so we have a big difference in the amount of light that's being received in the upper and the lower part here. And incidentally, this dotted line is showing approximately where the mixed layer might be during stratified conditions. And then we have also very low nutrient concentrations. Here I'm showing nitrate plus nitrite in the upper part of the euphotic zone, and eventually those do increase. And really the two of those properties together lead into the distribution of phytoplankton biomass and the development of what's called a deep chlorophyll maximum. So we have a sweet spot where nutrients are more available and there's still sufficient light for photosynthesis, and then we have this peak in chlorophyll fluorescence. On the other hand, that is not always the peak of where productivity occurs. In fact, most of the productivity is occurring shallower. Nonetheless, photosynthesis is occurring at the DCM and below. And so we can generalize these habitats of the upper part of the euphotic zone as a high light, nutrient limited habitat for photosynthesis, and the lower part as a light limited and high nutrient habitat for photosynthesis. And then we get this response from the photosynthetic community of producing lower chlorophyll in this upper part, so we see low chlorophyll to carbon ratios, and more chlorophyll in this lower part of the euphotic zone. So those are some basic observations that are very widespread. And then, as I said, these data are from the BATS site, and Station Aloha in the Pacific looks very similar. And I just pulled some data from a really nice recent paper showing how the primary production rates are distributed over the same depths. And indeed, the primary production is actually higher rates in the upper part of the euphotic zone and lower rates under these light limited depths. Nonetheless, if you integrate the total photosynthesis throughout the euphotic zone, this lower part is contributing on average somewhere around 20 to 25%. And what this recent analysis of the time series data from Station Aloha found is that, in fact, this deep photosynthesis in the lower part of the euphotic zone is actually more responsive to some climatological changes over the last several decades. So over these decades, we've seen an increase in primary productivity in this lower part of the euphotic zone and no similar detectable increase in the upper part. So this is one reason why it is certainly worth examining more closely what exactly is controlling the primary productivity in this lower part of the euphotic zone. So I have some other questions too, which largely relate to what gets exported out of the euphotic zone and how can we trace what has happened in the upper part of the ocean by what is sinking into the deeper ocean. Okay, so my work focuses around particulate organic matter, and the big question that I'm asking today is how does the composition of particulate organic matter reflect those biogeochemical differences between the upper and the lower parts of the euphotic zone, and also might affect the biogeochemical cycling in turn. And so when I talk about particulate organic matter, I'm talking about a big mixture of things. So when we collect particulate organic matter, or POM, we are capturing both the living and non-living components, unicellular components, and that involves both autotrophs and heterotrophs, photoautotrophs and chemoautotrophs. And with particles, there are a lot of physical dynamics to consider. So particles can sink through the water column, they can be produced as feces by zooplankton, and therefore get injected quickly at whatever depths the zooplankton are. And then we also have aggregation and disaggregation processes. So these are all things that we have to think about when we are trying to interpret the chemistry of what we measure in POM. And a couple other notes, what I'm going to be talking about today are some really specialized chemical analyses that reveal properties of POM and then biogeochemical processes of these parts of the euphotic zone. And also, we wouldn't really see what we're seeing if we did not do this at high depth resolution and also by collecting different sizes of particles so we can get at these dynamics. And so this involves some time-intensive sampling. So everything that I'm showing today was collected from shipboard sampling, and these are just some nice photos from a recent cruise I did just last month. Time-intensive sampling shipboard, labor-intensive analyses in the lab, and this is just something that I like to think about for the future. We're trying to get more global data through more autonomous platforms, and just thinking about in the future, what is the future of seagoing oceanography, and can we still support this type of discovery? Okay, so I'm going to focus what I show you today around 3 very simple observations and then going into depth about what we think they're actually telling us. So basically, by making measurements over and over again in different oligotrophic gyres, some patterns started to pop out to me that I did not think had been fully described. And part of that was because we had a lot of latitude within our programs to actually do sufficient sampling to capture these high-resolution depth profiles through the euphotic zone. So in some sort of classical geochemistry, you might just hit standard depths, 50, 100, 150 meters. You might actually miss these sorts of features. So very basic parameters here, the C to N ratio of the particulate organic matter, the bulk natural abundance delta C13 value of the particulate organic matter, and we're seeing some big differences between the upper part of the euphotic zone, which I'm delineating here with this line, and the lower part here, and then this is the base of the euphotic zone here. So we've got some big differences that we'll dig into, and then this is a more specialized measurement that I will be talking about as well. Okay, so some of the big questions that come from these very simple observations of sort of big unrecognized geochemical differences between the upper and lower euphotic zone is, do these differences in the habitats for primary producers, the high light, low nutrient, low light, high nutrient, create different organic properties in what is produced? Then, if we have different types of organic matter, do we have different sort of responses from the heterotrophic community? And then what does that mean for the overall biogeochemical cycling? So, and then as I said, I have a long-standing interest in what gets exported, what's available for the mesopelagic food webs, and then also what gets recorded in the sediments and how can we interpret it. I won't get so much into that today, but I'll try to touch on it. So first, this is going to be a story time order because this is the order that we actually sort of discovered these things. So I'll start with this pattern in the bulk delta C13 values of particulate organic matter. So the figure that I just showed was from the Bermuda site, and this is from some work that I had done previously at Station Aloha. Basically saw the same thing. And I will also just note here that this difference in the delta C13 value between the upper and the lower euphotic zone is huge. If you are, for instance, a geologist, you would think that this means some big shift in the carbon cycle if you were to see this in the sedimentary record. So to dig into this a little bit more, first we did a global analysis of whatever data were available out there. And so what we found was, we found this pattern over and over again in published data, and we found that this was global in the open ocean. So this was excluding ODZs, excluding places with a lot of riverine influence. So in open ocean locations, we saw this decrease in these delta C13 values between the upper and the lower euphotic zone, but what we also found was that there was no agreement about what caused this. And so the question going forward is this a essentially bottom-up signature coming from the photosynthetic community, or is this some aspect of degradation? And so just to add to our features of the upper and lower euphotic zone that I showed before, I've just slid in here some carbon parameters. So we see DIC concentrations, CO2 concentrations, and those increase between the upper and the lower euphotic zone. And then the delta C13 value of the CO2 that's used for the photosynthetic substrate also has a change over those depths. And so that is a consequence of the removal of CO2 by photosynthesis, removal of light CO2 driving this value heavy, and then respiration adding back in light carbon in the lower euphotic zone. So this is known. And so we suspected that what we were measuring in the organic matter was some consequence first of this pattern, and then some other features on top of that. So we really wanted to question whether there was a photosynthetic mechanism behind this. So what we did is we used compound-specific isotope analysis to isolate phytol, the side chain of chlorophyll, and that basically gives us an integrated picture of the whole photosynthetic community. If you would like to read more about compound-specific isotope analysis, I wrote a review about it several years ago. Lots of good stuff. So this is the work of my former PhD student, Lillian Henderson, and the bulk pattern that I showed you is just here. It looks a lot smaller because what we found in phytol, which we cleaved from chlorophyll, was a much bigger decrease in those delta C13 values. So to boil this down rather quickly, what we can do is we can look at the difference between those phytol values and the CO2 values, which that was formed from by the photosynthetic organisms, and we can calculate the fractionation during photosynthesis. And so this is something that people who work in the geologic record think a lot about because it can be indicative of the conditions that the photosynthetic organisms were living in. So here, by demonstrating this in a photosynthetic compound, we showed that the origin of this bulk pattern is in fact photosynthetic. And then we put this in the context of existing work, so thinking about the dynamics of CO2 uptake by phytoplankton and their growth rates. And we could partially explain what we saw. So we see that it fits nicely. These are our data in the bold symbols. They fit nicely into the same patterns of data found previously. But then if we account for also the surface area to volume of our phytoplankton community, we now see that there is something else driving what we're seeing. And what I will point out is that our study was really the first to look at this over depth. So most of the rest of this had been done in culture or looking at these variables over latitude instead of over depth. So something different is happening when you look at this over depth. And so Another broad scale thing I want to talk about today is the power of collaboration. So I am trying to feature nice photos of my collaborators, one of whom is right in front of me. So by having collaborators who are experts in phytoplankton community characterization, we were able to look at our data more in terms of the reality of what you see between the upper and lower euphotic zone. And so what we found, what we pulled out of that, is that there are factors that are not considered in a lot of these photosynthetic models, things like what phytoplankton are actually living at these different depths and under these different CO2 and nutrient conditions. And so the two things that we really pulled out that are not currently considered in these models is the community composition, and we found that small eukaryotes might have an impact on this, and then light itself. So usually in culture, the duration of light might be varied, but not necessarily the intensity. So we think the light intensity might have a factor here. Okay, so this we think is a potential tracer that we could use to examine whether material being exported out of the euphotic zone is coming from the upper or lower portion. So again, we don't really know the importance of that primary production in the lower euphotic zone to export unless we have a way of tracing the material, tracing the origin of that material. So we are still developing the possibility that these low delta C13 values could help us do that. Okay. So again, story time. We also realized while we were making these measurements, because we were doing all these estimates of the phytoplankton biomass as well as their delta C13 values, that we could do a mass balance calculation and also estimate the delta C13 value of everything that's not photosynthetic. So like I said, POM is a big mixture, and so we were able to calculate the delta C13 value of the other stuff, which would be heterotrophs and detritus. And so to bring this down to our conceptual figure from this paper, from our phytol measurements, we have the delta C13 value of the phytoplankton community. We saw that that decreased between the high light upper euphotic zone and the low light. And then here's our mass balance calculation of the other stuff. And basically what we find is there's a lot of similarity in the upper euphotic zone, and there's a divergence in the lower euphotic zone. And so what we think this is telling us is that in the upper euphotic zone, heterotrophs are living in a close association with the primary production, which fits with a lot of other theory that we think about in the upper euphotic zone. But as we get into the lower euphotic zone, we have accumulated detritus sinking from above, and so the heterotrophs living here can rely both on the in situ production and this sinking material. And so we get a divergence in the geochemical signatures of the photosynthetic and non-photosynthetic material at these depths. So this also then led us to question, well, what else might we discover about the heterotrophic metabolisms of the lower euphotic zone? Are there other things to look for that might shed light on what they're doing there? And so this leads us to this more complicated measurement, which is a compound-specific measurement for which we estimate trophic position. And I'll try to explain what trophic position means in particles. We measured the delta N15 values of individual amino acids, compound-specific isotope analysis of amino acids. And the basic theory here comes from animal food webs. So in animal food webs, the internal digestion of amino acids results in the increased delta N15 values of some amino acids of the consumer compared to its food, and no increase in other amino acids. So because that is such a predictable difference in that increase and no change across specific amino acids, this can be used to calculate trophic position. Again, this is developed in animal food webs. And so you can do something like this. You can relate the actual trophic position of an animal to the delta N15 measurements of individual amino acids, and therefore you can just use those measurements to estimate a trophic position. So how does this relate to microbes and POM? You have to think about what actually causes that isotopic enrichment. And so what causes that enrichment is there has to be some release of ammonium during a partial catabolism of amino acids. So by isotopic mass balance, the only way you can get isotopic enrichment in something like glutamic acid is if there's a net release of ammonium waste. So it is the light ammonium that is excreted, leaving the residual pool isotopically heavier. Okay. So if we think about it that way, that means that when we see an increase in trophic position, that means that somebody has done some catabolism of amino acids. When we're thinking about bacteria, however, heterotrophic bacteria don't have to do that. They don't have to act like an animal. And so this has been shown in culture, and it has also been shown not isotopically in culture, that if heterotrophic bacteria are growing on a nitrogen-poor substrate, they might not produce any nitrogen waste at all. Instead, they are essentially recycling the amino acids that have been provided to them, for instance, by the photosynthetic community. So what we found in the same exact samples where we measured that phytol is we were able to make that measurement, calculate a trophic position, and because we had a mass balance estimate of the heterotrophic or the non-phytoplankton component, we were able to estimate the trophic position of the material provided by the non-phytoplankton component. And what we saw is that increased over depth. And so what this boils down to is that as depth increases between the upper and lower euphotic zone, amino acids are being catabolized more in the lower euphotic zone, meaning they're being used for energy, whereas in the upper euphotic zone, they appear to be recycled efficiently and not used for energy. And so what that means is the implication is that there would be ammonium waste generated in the lower euphotic zone and not as much on a net basis in the upper euphotic zone. Okay, and we also found this in our one of our exports locations. So that was work by my former PhD student, Paul Voital. You can read that there. And so adding on to this picture, we now have this estimate of the trophic position of the material. And again, we're seeing that divergence in the lower euphotic zone. So again, a tight coupling between the photosynthetic and heterotrophic communities in the upper euphotic zone and divergence in the lower euphotic zone due to greater reliance on catabolism of the more abundant detritus at that depth. Okay, so then we thought about, now that we're thinking about ammonium release and that sort of thing, how does this relate to the broader biogeochemistry of the upper and lower euphotic zone? And then we get to this very simple observation of, but very robust observation of a decreasing C to N ratio between the upper and the lower euphotic zone. So this is continued work by my former PhD student Lillian, and what we found when we aggregated our elemental data from a bunch of cruises is we had these very beautiful profiles showing a very consistent pattern of high C to N ratios in the upper part of the euphotic zone, there's the dividing line there, and lower in the lower euphotic zone. And so our hypothesis was that this had to do with the biochemical composition of the organic matter, and with help from another collaborator in Craig Carlson's lab, we analyzed the carbohydrates in those very same samples, as well as the amino acid abundance. And what we found is that that varying C to N ratio, in fact, was largely driven by how much carbohydrate and how much amino acid was in these samples. And so what that means is we have a high production of carbohydrates in the upper euphotic zone and a higher proportional contribution of amino acids in the lower euphotic zone. So putting that together with what we were just talking about, that means that in the upper euphotic zone, there seems to be exactly that situation where heterotrophic bacteria would be metabolizing nitrogen-poor organic matter and not necessarily producing a Redfield balance of nitrogen waste. And so putting this side by side, this was our trophic position of our non-phytoplankton parts of the POM. Our hypothesis is in the upper part of the euphotic zone that amino acids are going to be recycled for cell growth instead of catabolized for energy. And then in the lower part, the carbon is going to be oxidized for energy, the nitrogen is going to be expelled as waste, and this is also going to provide fuel for nitrification, which takes off in the dark right around the base of the euphotic zone. And that's going to produce additional oxygen demand during that oxidation as well. And so I just showed you this in a cross plot, but here are the depth profiles of the carbohydrate abundance and the amino acid abundance. And so we do see that higher carbohydrate proportion in the upper euphotic zone. And so we hypothesize that these are being used more for energy, along with energy supplementation through photoheterotrophy. So this is kind of the overall hypothesis that has come out of these detailed geochemical observations, that there's community nitrogen limitation and catabolism of abundant glucose in the upper euphotic zone, and in the lower euphotic zone, more of that amino acid is available for energy usage, and that produces that ammonium waste. So this is our hypothesis, and now we get to test it. And so with further work from the Bioscope project, which you can read more about there, we've been digging into this. So this is work by Jackie Comstock, looking at the heterotrophic prokaryotic communities. And I'm highlighting here the depths that I was just talking about. So the upper euphotic zone communities are really, really distinct from the lower euphotic zone communities. So we're not necessarily looking at their metabolism, but we can see there's definitely different communities there. To look into those metabolisms, we're starting to look at the metagenomic capabilities at the different depths. We can look at their proteomes with aforementioned max cyto. We're looking to do some cell sorting techniques and also to make sure this makes sense with what we're seeing in the DOM pool as well. And then there's some broader implications that we're interested in looking to in terms of the stoichiometry of remineralization. As well as what that means for what nutrients are available for photosynthesis, and then what models are going to be affected. So models, for instance, that assume the Redfield ratio are going to be highly affected by these findings. And the continuing work on this project in my lab is by Dalen Jing, so look out for more of that. To put this all together, we layered this carbohydrate data on top of the isotopic data that I described before, so more glucose up in the upper euphotic zone and low carbohydrate in the lower euphotic zone. And I'm just going to put those up really quickly, our hypotheses. And just also note that the implication of our hypothesis here of greater ammonium release during amino acid catabolism in the lower euphotic zone would also mean fuel for ammonium oxidizing archaea. So that leads us now to what is exported. So what's exported to the deep ocean to fuel the food webs there? What's exported to the sedimentary record? The pictures I put up here are to remind me to talk about mechanisms of export. So this POM can get exported if it aggregates into large particles that can settle gravitationally, and also if it is consumed in the metazoan food web and packaged into feces. So as we think about what is exported, we also need to think about where zooplankton are feeding in the water column and where aggregation is occurring. And so the question that you can think about, even if you work in other types of stratified systems, is if you have geochemical differences, say between the upper and lower euphotic zone or a chemocline or something like that, what are the export processes that are layered on top of those differences? And which of these signals is actually going to get into your deep ocean food web and into the sedimentary record? So some of the ways that we're starting to look at that, one of my former graduate students, Shannon Doherty, used amino acid compound-specific isotope analysis to essentially develop a signature of fecal pellets that's distinct from phytoplankton aggregates or microbial biomass, and therefore might give us a tracer of where fecal pellets are being produced in the water column. And so this is just an example from Jackie's paper that I brought up before, where we found in these large particles in red, my depth axis got cut off, sorry, this is the base of the euphotic zone, so this is in the lower euphotic zone. What we found is a high trophic position and these low delta N15 values of threonine, both of those indicate that fecal pellets are showing up in the lower euphotic zone specifically. So this is giving us the idea the lower euphotic zone actually might be a hotspot for fecal packaging and therefore might actually efficiently package all of those geochemical signals from the lower part of the euphotic zone. We also, through these methods, and because we look at size fractionated particles, we get signatures that might relate to particle disaggregation. So what we see here, just below the euphotic zone, is those same fecal pellet signatures showing up in our smaller particles. So we're also starting to dig into whether right below the euphotic zone is a hotspot for particle disaggregation. So we kind of need to think about all of these when we think about what is making it to which depths of the water column. And there's a bunch of results coming from the exports program around this type of categorization of particles. One is published by my former student, Paul, and there's a couple in the works from my former student, Liz. So be on the lookout for those. And then also, two of my new projects are also really digging into this question. Now that we are developing these really different signatures from different parts of the upper water column, how can we track those into what is exported so that we can understand better how production in the lower euphotic zone actually translates to carbon sequestration or to the sedimentary record? And so these are two projects with the Schmidt Sciences Ocean Biogeochemistry Virtual Institute. If you haven't heard about it, it's 5 really big collaborative projects. So I'm just showing a couple of my collaborators here. So one of these projects, we are basically expanding our characterization of feces to all kinds of animals, all kinds of depths in the ocean to get a better signature of where fecal packaging is occurring. And then the Subsea project, which is headed by Matt Church at University of Montana. This is explicitly focused on the lower euphotic zone and its importance in biogeochemistry. So we have a bunch of collaborators, a lot of people in Hawaii associated with the Hawaii Ocean Time Series, and we are really, really digging into the specific nutrient budget of the lower euphotic zone. So be on the lookout for work from those as well. And I will just acknowledge a bunch of people from my lab over the years who have contributed to this work. We do have another new project that is looking into the fundamental aspects of isotopic fractionation during these bacterial amino acid metabolisms, and I am currently advertising for a postdoc. And then thanking my funding, largely from the Bioscope program from what I've shown you today. So I think I have a few minutes for questions if anybody has any. Thank you. And also come talk to me about Lake Erie fertilizers at any time. Yeah.
Hi, thank you, an amazing presentation. And in the subtropical gyres, the upper part is dominated by picocyanobacteria, Prochlorococcus and Synechococcus. And in the DCM and below, there's an increase in eukaryotic algae. And cyanobacteria accumulate mostly polysaccharides as a reserve energy, while eukaryotes can shift to accumulate liquid droplets, for example. Is there any effect in the isotopic fractionations due to the upper dominated cyanobacteria community and the lower eukaryotic dominated community?
Yeah, so that's a great question. In terms of the cellular mass balance, basically all the CO2 that comes in, there has to be an isotopic mass balance to all of the biochemical products of that organism. So the way that we think about it right now is that the net biomass is still going to resemble, it's going to reflect the isotopic fractionation during photosynthesis. The thing that I think might really affect that, which nobody is looking at, is how much of that primary production actually goes into the dissolve phase. Because if there is dissolved production that we are not capturing, then we don't have an actual isotopic mass balance. So that is one of the things that we mentioned in our paper, that dissolved production is a big variable. We didn't see at our study site a huge difference in the lipid composition, but another thing I think that is a huge lever on this is the expression of carbon concentrating mechanisms. That's one of our, basically another big lever on that isotopic mass balance. So I think this is largely a question for people who are trying to make really great models of carbon isotopic fractionation, and that is largely why they don't exist in a perfect way just yet. There's a lot of these variables. But yeah, I think especially looking at that really high carbohydrate content in the upper euphotic zone, we really want to dig into like what form those carbohydrates are in actually. I think there was a question.
Thank you very much for such inspiring talk. It was for me nice to see that the amino acids that are recycled in the upper photic zone can be attributed to the photoheterotrophic organisms. And the question is, did you manage to decipher what type of photoheterotrophy is the one driving this effect, since the two main types might have different nitrogen requirements?
Yeah, so the quick answer is no. That's basically next steps is looking into how photoheterotrophy could play a role. I think a really interesting thing that we can do is try to put together an energy balance, basically, for, if we think about the light availability at different depths, can we include that as part of the energy balance of heterotrophy? But I guess another thing to mention is we're starting to work on incorporating biomarkers for photoheterotrophy, like proteorhodopsin, as yet another marker that we can use to attach to these different processes. But yeah, to come in the future, I guess. Else? If not, thank you so much.
Okay, with that, I want to thank Drs. Bonilla and Close once again. I want to remind all of you about the rainbow reception, and thank you all for being here. Good night.